Hello, we are Group 18, or as we jokingly call ourselves, the Dumpster Divers. Our group's concept was to reduce waste by using waste, but also by using less. For this goal, we decided not to use any 3D printed parts. We mainly got custom parts by scavenging the courses and other groups' waste. We used zip ties wherever we could, and most of our parts are loose, as we didn't want to create waste by making proper holes. Our main innovation was the monstrosity you see on the screen now. It is a pump repurposed to act as a pulley. All it took to make it was the DC water pump, an empty reel we found laying on one of the many tables in Makerspace, a small part of thick tubing that we used to attach the reel to the motor, also found in Makerspace, some borrowed duct tape, and a string we had laying around. This pump, now pulley, was then programmed to let us switch between oxygenating the water or feeding the mussels. The mussels bucket is placed in a cardboard box to reduce as much light pollution as possible. Our OD sensor is pretty sensitive, so we have to get the lighting conditions as stable as possible for it to work smoothly. Looking at the video, the wiring might look like a fine dish of spaghetti, but unlike spaghetti, we made sure the electronics are separated from the water. Furthermore, we made sure no wires would disconnect from a little bit of shaking. Let's talk about the elephant in our bucket-sized room. The OD sensor. Our OD sensor is, as we talked about in the last review meeting, a red LED, a violet LED, and an infrared sensor capable of also detecting visible spectrum light. What we didn't expect from last time was that we also needed a stronger light, so we used the algae lamp. To calibrate the sensor, we took OD measurements at 10 different known concentrations of algae. For each of these concentrations, we measured 10 times each and used the mean to fit an exponential function to it. Question everything is the motto for our biology part, so we did. We divided our materials into two groups, the ones touching the water and the ones not touching the water. We found out pretty quickly that the materials not touching the water are harmful to mussels, so we made extra sure that they definitely would not touch the water. A big scare we had was the string we used. Apparently, mussels are harmed by microfibers, especially the ones string is commonly made of, polyester and cotton. To fix this issue, if the project went on longer, we would change our string to a silk string. Well, enough the bio part. Lar, let's talk energy gains.
For the energy part, our design uses a stepper motor and one small water pump instead of two air pumps, reducing the energy use. Over 24 hours, our model consumes just 56.5 watt-hours compared to 67.62 watt-hours for the default setup. That's a 16% energy saving with fewer components.
We managed the model using our parameters, which allowed us to find how much the mussels need to decrease the concentration of algae for each possible range of algae concentration. With this, we found the optimal concentration, which is almost at the point of the maximum feeding rate. We discarded the original idea and went with a simpler approach, as the siphon turned out to transfer water much more slowly than a pump. Additionally, we set the minimum feeding time to be more than half an hour, so as to have the time to oxygenate the water. This resulted in the optimal starting concentration to be at around 5,000 cells per milliliter.
For the remote monitoring and export of the experiment data part, the idea is to store the data on our laptops and on the HUSA 32 board, where we will access it via its Wi-Fi chip. This would mean that we can control things like the cooler status, the pump status, and we could also see the concentration of our algae as in the picture. However, a unique thing to us is that we can control and set the temperature of our mussel tank on our dashboard, so we can change it to be any temperature that we wish, and which allowing us for greater control of the mussel tank. This slide shows how we tuned and tested our cooling PID controller. We used proportional, integral, and derivative values of 8.5, 3, and 0.5. The cooler pump power is scaled directly by the PID output. As you can see from the graphs, after an initial drop, the system stabilizes quickly, both in terms of temperature and pump power. We used trial and error to find the PID values, and they resulted in a stable performance and in a consistent temperature control. Here, we show 3 additional measurements. First, we map the motor power versus the water flow rate in order to see if we would lose water. Then we tracked the algae growth, which mostly followed our expected 1.65 rate, which means that we will not run out of algae. And finally, we monitored the algae feeding over the time, which steadily decreased as it was expected.